I'd like to welcome you all to today's Wednesday seminar, and I'd like to begin by celebrating and acknowledging the traditional owners and custodians of the land on which we meet today, the Wurundjeri people of the Kulin Nation, and I pay my respects to their elders past and present, and I embrace their continued connection to this place. Now, it's my great pleasure today to introduce our speaker, a very close and valued colleague and co-head of the Immunology Division, Daniel Gray. Daniel's scientific interests and journey combines topics very close, I believe, to the beating heart of WEHI. These are the thymus, cell death, immune regulation, and even lymphoma. Daniel first learned to love the thymus in Richard Boyd's lab at Monash in the early 2000s, and this was further enhanced by successful postdoctoral research in the prominent lab of Diane Mathis and Christophe Benoit at Harvard, where he studied the autoimmune diseases caused by air-deficient mice. From there, he was recruited to WEHI and joined Andreas Strass's lab for a few years before he was appointed to lab head in 2013. In 2019, he stepped up to his current role as division head in immunology. At WEHI, Daniel has explored the intersection of cell death and survival on T-cell control, making many major discoveries, particularly on the regulation of FOXP3 Treg cells. Recently, his lab has been working on a number of new directions that open up new insights for old problems, particularly around thymus involution. And Daniel, we very much look forward to hearing this update. Thank you.
Thank you, Phil, and thank you all for coming. So what I hope to do today is to detail two studies, two recent discoveries from the laboratory. One about this curious feature of the thymus, which is that it undergoes age-related atrophy, referred to as thymic involution. So this is really an old problem. And the second study is about the wiring of cell death processes in a special class of T cells, regulatory T cells, and distinctions in those wirings that allow for fine control of T cell responses. These stories sort of fit within a central theme of the laboratory, which is to understand how the immune system makes decisions and can distinguish our own body from potentially harmful pathogens. And so this is an essential decision-making process because the immune system constantly has to maintain a balance between immune responsiveness on one hand, to cause inflammation and clear pathogens, versus the imperative to maintain tolerance of our own tissues. And tolerance is a state of non-responsiveness, so the immune system tolerating our own tissues. This balance is critical in a number of disease contexts because if you upset this balance, for instance, in favour of tolerance, you compromise the immune responses too much and weaken an individual's ability to resist infection, and that can drive immunosuppression. Conversely, if you tip the balance in favour of inflammation, then this can result in inflammatory pathology that can cause a range of diseases such as autoimmune conditions and allergy. And so it's this balance that clinicians seek to redress when treating autoimmune diseases and allergy. Likewise, the same decision-making process on the left here is the one that cancer immunotherapies target to improve immune responses to cancer. And it was through fundamental research into this process by Jim Allison and Tasuku Honjo that discovered CTLA-4 and PD-1 as central regulators and opened up immunotherapy as a new way to interfere with this balance and provoke immune responses to altered self in cancers. So there's many dimensions to this balance, but we have a particular focus on T cells because they're the ones that orchestrate immune responses to viruses and autoimmune diseases, but also have particular relevance for immunotherapy of cancer. So it'll be helpful to know where T cells are made. And all the blood lineages arise in the bone marrow with the exception of T cells. They're special. They require a specialised organ for this to happen, and that's the thymus. And it's shown here. It's this bilobed organ sitting atop the heart. And this is the reason they're called T cells. They're thymus-derived lymphocytes. And Jacques Miller made this discovery many years ago. And he doesn't get nearly enough credit for that, nor the subsequent work he did here at WEHI, identifying the division of labour between T and B cells. This was all foundational for the discovery of immunotherapy, vaccine design, many drugs. This is a story for another time, but it was a really important discovery that the T cells require a distinct organ for their development. Suffice it to say that ever since this discovery, the question of how T cells are made in the thymus and the way immunological tolerance is imposed in it has fascinated immunologists for decades. And this schematic here on the right shows a thymic lobule and the key microenvironments that direct T cell differentiation. The two major zones are the outer cortex and the inner medulla. And the process begins when bone marrow-derived progenitors arrive via the circulation in through large blood vessels at the junction between the cortex and medulla. And once in the thymus, the progenitor cells move into the cortex and receive signals from these specialized epithelial cells, cortical epithelial cells. Sometimes I'll refer to them as TECs, thymic epithelial cells. These provide signals that commit these progenitors to the T cell lineage and drive proliferation, like impressive proliferation. A handful of progenitors every day gives rise to billions of developing T cells in the thymus. And this is important because over 95% of those cells are destined to die in the thymus, which seems rather wasteful, but I tell you why that's important. The reason for this is that during the early stages of T cell differentiation, there's a process by which the TCR, the T cell receptor, genes are randomized. Now, the TCR is the primary way that T cells sense their environment and sample for potential threats. Therefore, it's of critical importance to this decision-making process for tolerance and immunity. Now, the recombination process essentially randomizes the TCR specificity, that is, the types of shapes that the TCR is capable of interacting with. And I've represented this clonal diversity here with all the different colours of the individual T cells. Each person has 10 to the power of 11 T cells with distinct TCR specificities. And even this vast number is only a sample of what possible TCRs can be generated through this recombination process. So this prodigious breadth means that the immune system's capable of recognising virtually any shape, any threat, even pathogens that have never existed before, such as SARS-CoV-2. We have T cells circulating around it, have TCRs that can interact with it. So that's great, but there's two problems with this random process. The first is that many of the TCRs that are generated are not capable of initiating an immune response. They're not capable of interacting with the way the immune system displays these peptides. And so they would be useless. Now, there's specialized CTECs, the cortical thymic epithelial cells, drive a process, a screening process called positive selection that causes any of these useless cells to die and selects only the ones that are capable of interacting and potentially causing an immune response. They advance into the thymic medulla. Now, in the thymic medulla is where a second process occurs, referred to as negative selection, and that deals with the second problem posed by random TCR gene rearrangement, which is that by chance, some of these TCRs will have a very high interaction with self-constituents. And if that happens, if those cells were to mature and be exported into the peripheral immune system, they could launch an autoimmune response. For instance, insulin-specific T cells that might cause type 1 diabetes. So it's really important that there's this second screening phase, and it occurs in the thymic medulla. And what happens here is that MTECs now, the medullary thymic epithelial cells, provide a preview of peripheral self to the developing T cells. And any that should interact with peripheral self with too high an affinity are directed to undergo cell death. Now, there's two extraordinary mechanisms that operate in MTECs to broaden this scope of thymic tolerance and expand our cast of characters, if you like, that are in this stage. The first involves the transcription factor that Phil mentioned before, the autoimmune regulator, or AIR. This transcription factor directs the expression of thousands of genes in medullary thymic epithelial cells. And these are genes normally found only in peripheral tissues, and maybe just a few. Things like insulin, thyroglobulin, salivary proteins, you name it. They're directed by AIR to be expressed by some of these medullary thymic epithelial cells. When they're ectopically expressed by these MTECs, they're shown to the developing T cells, and if they should interact with too high affinity, they're killed. Now, as if that's not enough, it's recently discovered that some of these MTECs adopt distinct cell fates that recapitulate the transcriptional program of peripheral lineages, like cells normally only found in the gut or the lung, for instance. Things like tuft cells, microfold cells, ionocytes, muscle cells, you name it. There's up to 15 different lineages that are recapitulated in the thymic medulla, and they're collectively referred to as mimetic epithelial cells because they're mimicking the programs and morphology of cells normally only found in the periphery. So this is a remarkable representation of self by at least two independent mechanisms that can broaden the scope of immunological tolerance, and emphasising how important it is to know thyself, that there's these mechanisms in the thymus to impose immunological tolerance. Now, the minority of developing T cells that make it through these stringent selection processes, then complete their maturation, are exported into the periphery and circulate as T cells, patrolling now for potential pathogens. Thus, it's the distinct epithelial cell types in the thymus that orchestrate the processes of thymocyte differentiation, and no other cell type in the body appears to be able to do it. So it seems weird then that the thymus should be the first organ in the body to undergo age-related atrophy, referred to as thymic involution. No one knows quite why this happens, but the consequences are clear. The lymphoid tissue in the adult thymus is replaced by adipose tissue, and you lose a lot of the structural integrity that's important for driving the processes of T cell differentiation. This graph on the right here measures newly produced T cells being exported, and note the log scale, and you can see that shortly after birth, the number of T cells made by the thymus and exported is diminished greatly, and this accelerates after adolescence due to the action of steroids. And this phenomenon is known to occur in any animal that's been measured to have a thymus. So the reduced production of new T cells has been linked to the poor immunity of aged people, particularly when confronted with new pathogens, seasonal flu, SARS-CoV-2, you think of those. And this is because the T cell repertoire in all the T cells in that person is diminished, because as they accumulate responses as they go through their life and accumulate memory T cells to maintain immunological memory, this comes at the expense of untrained or naive T cells. And because the thymus isn't making new ones, our ability to respond to new pathogens wanes. This has prompted the question why thymic involution happens, and there's been several interesting hypotheses put forward because no one knows the answer yet. The first one is that it's important to conserve energy because this production and killing off of so many developing T cells every day would seem rather wasteful. So the dispensable soma hypothesis was put forward. Another is that it reduces the risk of malignancy or autoimmunity because these are error-prone processes. Think of acute T lymphoblastic leukemia and autoimmune diseases as the thymus is losing function and structure, that perhaps involution diminishes that risk. Another put forward by Jacques himself was, well, it's simply dispensable. It doesn't matter. It's not applying a selective pressure because people are well past their reproductive age before it becomes a major problem. I like that one. Another one is that maybe it's important to make space for memory T cells, and they're the ones you want because if you've been confronted by a pathogen once, the chances are you'll see it again. So maybe that's the balance that needs to be redressed. And of course, Phil and his colleagues also put forward another idea, which is that it's required to optimise the selection of T cells in the peripheral homeostasis mechanism. Regardless of the reason, there's a severe and clear impact of thymic involution in the setting of myoablation, followed by hematopoietic stem and progenitor cell transplantation. And this is for the treatment of certain blood cancers. It's essentially a reboot of the immune system, if you like, and a critical parameter of success for these is how quickly people can reconstitute their immune system. And in this representation of the recovery of various immune cell populations shown here, you can see that patients in the yellow line quickly get back granular size, often supported by GCSF. Most B and dendritic cells come back relatively quickly, but you'll appreciate in this red line here, the T cells come back very slowly. They take years, and in some patients, they never really are able to restore T cell immunity. And this in part is because the thymus has involuted and been damaged by that myoablative therapy. Children tend to do better with these treatments, and that's correlated with a swifter recovery of T cells because they've still got good thymic function. So what's required is a way to somehow restore thymic function and reform the T cell pool. So the questions that motivated this first study are, what are the major changes that occur during thymic involution that might drive this process? We'll be particularly focused on the epithelial cells of the organ, because as I mentioned, they're the ones that govern the critical processes of T cell differentiation. And secondary to that, what what's changed in the involuted thymus that impairs the regeneration of the thymus. As I mentioned, the thymus can regenerate, and if there's an acute insult that causes brief atrophy in a young person, is shown here in the yellow line, the thymus will recover back to sort of normal steady-state levels of T cell production. But if that same injury confronts an aged individual, the thymus doesn't ever seem to be able to get back to that baseline. So while prior studies had quantified the general loss of thymic tissue and some of those structural changes, we lacked a systematic way to measure epithelial cells in situ to understand what's going on during thymic involution. Now, we were inspired by some of the imaging approaches taken by Edwin Hawkins and Joe Groom to look at bone marrow and lymph nodes. So Julie Sheridan, together with a talented PhD student, Kaylin Zhao, started an amazing collaboration with the CDI, Kelly Rogers, and in particular, Verena Wimmer. And the goal was to resolve the numerical and structural changes in epithelial cells in these different settings. However, tech provide a few technical challenges that have held back the field for years. The first is they're relatively rare, less than 1% of the total thymic cellularity. Secondly, they form a really complicated 3D network, as shown here in cyan, representing the cytoplasmic fluorescent reporter that's only being expressed by text. And the magenta cells are these developing T cells that sort of percolate through this network of thymic epithelial cells. Now, as I mentioned, there's many different types of thymic epithelial cells, so it can be hard to resolve that heterogeneity. And finally, it's really hard to isolate these cells in a way that keeps them alive. They don't like to come out of the thymic microenvironment, so it's unable to really study them in vitro. So one way that Julie and Kaylin tried to overcome some of these issues was to create a new tech reporter line where the FOXN1 promoter drives expression of the Cre recombinase. FOXN1 is a critical thymic epithelial cell transcription factor expressed only by text in the thymus. And this would switch a dual reporter system. So instead of expressing a TD tomato reporter, which all cells in this mouse do, they now express a GFP reporter. And an important twist is that both of these reporters have a nuclear localization signal. So they're only expressed, GFP is only expressed in the nucleus of the thymic epithelial cells, whereas all other cells will express TD tomato in their nuclei as well. And you can see with confocal analysis, the GFP signal shown here in cyan, TD tomato in magenta, Kaylin's able to nicely resolve individual thymic epithelial cells. And she was able to confirm with other markers that we have a higher, as expected, we have a higher density of thymic epithelial cells in the medulla, that place where negative selection happens. And while there's a lower frequency in the cortex, remember these cells send out these broad processes and link up to make this mesh work. So having confirmed this reporter worked as it should, Kaylin was then able to prepare and optically clear entire thymic lobes from mice and subject them to light sheet imaging. This produced some spectacular results. So here's a thymic lobe from a young FOXN1 NTNG mouse. And again, the GFP signal in TEC is shown in cyan. The medullary regions are most prominent features you can see because, as I pointed out, they have the highest density of thymic epithelial cells. If we create a surface around this medullary region now, in blue representing those that are in contact with other medullary areas, and in yellow those that form little isolated islets, you can appreciate that the majority of the thymic medulla in young mice is interconnected. We have evidence that it follows the vascular tree, but it was a surprising feature to us because if you look in 2D, they're always just these little round sort of blobs. Now, it's a very different picture when Kaylin looked at the involuted thymus from a 2-year-old FOXN1 NTNG mouse. Here, there was a striking new feature in this very high-density GFP signal that you can see, these band-like tissue that's now stretching around the thymic medulla. Now, the medulla shown here in magenta, it also regressed into these little blobs. You'd lost that interconnected structure that was typified the young thymus. However, we're most struck by this high-density GFP signal. It's something you simply did not see in the young thymus. So then Kaylin took a closer look at this by confocal imaging of 200 micron thick sections. And on the left, you can see the nicely distinct cortical and medullary regions of the young thymus. Although these microenvironments are present also in the 18-month-old thymus, you can see the addition of these high-density thymic epithelial cell regions. And this striking because the thymic epithelial cells seem to be really tightly packed together, which is in contrast to this three-dimensional structure that the thymic epithelium normally makes. consistent with what we saw in the light sheet imaging, they tend to be associated with medullary regions. And when Caitlin first showed me this data, I'm a skeptical type of person, right? So I'd spent many hours looking at thymic sections throughout my career, and I'd never seen anything like this. So I immediately thought, artifact. There's something wrong. Maybe what's happened is you've got activation of the GFP allele in a mesenchymal population, like fibroblasts are known to be an important part of the microenvironment. Fortunately, Caitlin's stubborn, or should I say rigorous, and so she then went and took a whole lot of markers to mesenchyme and endothelial cells and epithelial cells and proved me wrong. So here's the best example of many, where she assayed these high-density TEC structures. So she's this dotted line here demarcates one of these high-density epithelial regions, and she stained them with pan-keratin. Keratin's a key feature of epithelial cells in all tissues, and you can see that these structures light up beautifully for keratin, a central characteristic of epithelium, establishing that high-density TECs are indeed TECs. She then wanted to know how quickly they emerge and when this unusual microenvironment begins. And you can see here that while they're not apparent at 8 weeks of age, they're beginning to form in the involuted thymus of a 1-year-old mouse. And this continues even into 2 years. So there's an expansion of it even as the thymus loses further size. And that's just quantified here. Thymic lobe volume decreases both in the cortex, particularly that medullary microenvironment, the one that's important for immune tolerance. However, these high-density TEC regions are apparent by 1 year of age, and if anything, they trend upwards even as the thymus gets smaller. Now, around this stage, Caitlin also noticed a preprint on bioRxiv from a group that reported a single-cell RNA sequencing analysis of thymic stromal cells taken from young mice, but also from 18-month-old mice. And this piqued our interest because we predicted that these high-density TEC regions, that the cells composing them should be captured in such an analysis. And so in this analysis, led by a talented bioinformatician, Anastasia Kusa, demonstrates it's a UMAP visualization of these single-cell data sets. And in blue, she's showing the cells from a young thymus, and in red are the ones from the old thymus. And they cluster into well-known stromal cell populations like fibroblasts, endothelial cells, and here are the epithelial cells down here. And you'll appreciate there's more of these red zones that are apparent in the epithelial areas, indicating that the biggest changes are occurring in the TEC compartment. So she took a closer look here and sort of subfractionated the epithelial clusters and identified a range of previously described epithelial cell types, like the cortical epithelium, these various MTEC populations, including some of these mimetic populations, the tuft ones, goblet cells, etc. But she noticed there was 2 distinct features, ones that did not have a transcriptional signature that correlated with anything that had been reported before. And that's shown on the right-hand side here. And you'll appreciate it overlaps with this region where only cells from the 18-month-old thymus occupy. So they referred to these as age-associated TECs, or AA TECs, and there seem to be at least 2 populations. So we were interested to know whether these might correspond with the high-density TEC regions. And consistent with their location in the UMAP, they did a survey of genes characteristic of all these different subsets, the medullary and cortical epithelial cells, and found that by and large, these AA TEC populations didn't transcribe any of these genes. So sort of confirming the fact that they're a distinct cell population. And curiously, some of what we might think of as canonical epithelial cell features, like the expression of epithelial cell adhesion molecule, EpCAM, was lost in the AA-TEC2, which was a really curious feature that I'll expand on in a little while. But to try and unify the data sets that we had, we turned to a couple of different approaches. And one of the first pieces of evidence that they're the same things is shown in this spatial transcriptomic analysis of thymi taken from a 2-month-old mouse or an 18-month-old mouse. And this is done on the Visium platform. In the H&E stain section here, you can discriminate cortex and medulla based on the density of thymocytes in those 2 regions. And in pink, we're showing the medulla versus cortex in the green. And if you then superimpose a transcriptional signature that typifies AA-TEC1 or AA-TEC2, you'll appreciate there's very little signal in the 2-month-old thymus, but you can appreciate some of it in the 18-month-old thymus now. And notably, they're kind of associated with these medullary regions that we would have predicted from our imaging studies. And also, interestingly, they seem to be occupying the same region. So we would postulate that AATEC1 and 2 are both caught up in these high-density TEC regions. In another approach, Kalin undertook a careful analysis of the differentially expressed genes in AATEC1 and 2 to try and find potential markers that she could test as candidates by imaging. She found and tested many of these, but the most definitive was claudin-3. So this is the transcriptional profile of claudin-3 here, and we're highlighting these AATECs that all of them seem to have a very high transcription of claudin-3. It's known that some medullary epithelial cells also express it, but they're relatively rare. So when she took an antibody and stayed for claudin-3, shown here in yellow, and again we're identifying these high-density TEC regions, and we've demarcated it with this dotted line, you can appreciate there's this beautiful overlap between the reactivity of this claudin-3 antibody and these high-density TEC regions, shown in a couple of examples here, as distinct from those few scattered cells you might find in the thymic medulla that express claudin-3. So that was nice confirmation to us that these high-density TEC regions incorporate AATEC1s and 2s. And with this information, we could more precisely phenotype the age-associated TECs at the single-cell level. Kalin isolated thymic stromal cells from young and aged reporter mice and gated on the non-hematopoietic stroma from these cells. You did an enzymatic digestion protocol to liberate these cells. Now, we can identify all thymic epithelial cells on the basis of GFP expressions. This is any cells that have a history of FOXN1 expression. And so if she does that and gates on the majority here from the young mouse that express EpCAM, there's two other markers that we can use to distinguish cortical epithelial cells, marked by LY51 on one hand, or medullary thymic epithelial cells, marked by UEA1. And this includes all those populations I outlined earlier that live in the medulla. Now, this picture changes in the involuted thymus. While the majority of cells still express EpCAM, if we look at LY51 and UEA1 expression, we find the emergence of a new population, an aberrant population that doesn't express either of these two markers. And this population stains with that claudin-3 marker I introduced earlier on, confirming that now we've got the emergence of these age-associated TECs, that AATEC1 population at the single-cell level. And then, in addition, we find there's a new population that's GFP positive, so it's a thymic epithelial cell, but doesn't express EpCAM. And you'll remember those plots I showed earlier where that AATEC2 population we suspected was no longer making EpCAM. And indeed, that's the case. If we look at differentially expressed genes in the AATEC2 population, we find podoplanin is upregulated, and this population nicely stains with podoplanin here, distinguishing it from fibroblasts and other cell types that we can identify that express podoplanin in the thymus. So these data confirm the AATEC phenotypes and provide markers for other investigators now to use to interrogate these cell types without having to use the fancy reporter that we generated. Now, in the process of analysing these images of AATECs, Kalin noticed that they typically had very little TD tomato signal. So if you look here, again, the AATEC regions are shown here, and you can appreciate that the TD tomato signal is much dimmer inside of them. And now the thymus is typically packed full of developing T cells. You'd be very hard-pressed to find a region that doesn't have them. But this suggested that perhaps they're missing from this microenvironment. And further evidence of that came from, again, this spatial transcriptomic analysis, where we're looking at an aged thymic section now and highlighting again the AATEC1 and 2 signatures in these warm colours. And then if we look for transcripts associated with developing T cells, Thy1 and LCK, you can appreciate that they're missing from these regions that have high concentrations of AATEC1 and AATEC2, suggesting that AATEC form a unique microenvironment that excludes developing T cells from it. So of course we were curious to know if this is a feature of the human thymus, so Anastasia analysed publicly available single-cell RNA-seq data of TEC, which included data from a 35-year-old donor. And the AATEC1 and 2 signatures were able to be detected, indicated by this red colouring, and get AATEC2 and 1 popping up in this cluster, and also perhaps a distinct cluster of AATEC1 signature on the right. And if we look histologically now, this is thymus sections taken from a 50-year-old female donor. In this H&E section, you can see there's this prominent area with pink staining, with epithelial morphology that seems to exclude the purple staining lymphocytes as they're developing. We can confirm this is made up of epithelium using a pan-cytokeratin marker showing here. And then the exclusion of developing T cells is typified by the lack of staining with CD1A. This is a good marker of developing T cells in humans. So these are features consistent with the detection of AA-Tex that we found in the mice. And so just to summarise this bit, 3D imaging was critical for revealing this aberrant microenvironment in the involuted thymus. It's typified by an unusually high density of thymic epithelial cells that excludes developing T cells. So at the very least, it's not supporting the normal function of the thymus. And single-cell and spatial transcriptomics revealed they're formed by at least 2 populations of AA-Tex that have lost key characteristics of thymic epithelial cells. So to understand more about those characteristics, we did a closer survey of epithelial cell functions and whether they're represented in these AA-Tex. And these include regulators of T cell commitment and proliferation, so these are CTEC functions, but also negative selection and the pruning of the repertoire of potentially autoreactive cells. And in general, the AA-Tex populations expressed none of these genes, suggesting that they're unable to support T cell differentiation, consistent with the fact that you don't find T cells in those regions. A broader gene set enrichment analysis confirmed that defects in antigen processing pathways, so this is the mechanism for driving positive and negative selection events. The dark bands indicate a ranking of the genes, and you can see that those associated with this function sort of cluster with all the other thymic epithelial cells, but not the AA-Tex ones, indicating they've lost that ability. And likewise, we saw the emergence of a curious epithelial to mesenchymal transition signature in the AA-Tex2 population. You can appreciate many of these genes are highly expressed by AA-Tex2s, but not the other cell populations. This included that loss of EpCAM and E-cadherin, another epithelial marker. There was transcription of vimentin, but it seemed they hadn't gone all the way because, as I showed you before, they're still expressing cytokeratin. This raised the question of their origin and fate. And for this, Anastasia turned to RNA velocity analysis. She takes a single-cell RNA sequencing data and uses a method to infer their temporal dynamics based on mRNA splicing. And so here we've got the single-cell data set from the 2-month-old thymus, and the arrows indicate the predicted direction of the differentiation of the cells. And you can see that most of the medullary epithelial cells are derived from a highly proliferating subset here. Some switch on air and direct these processes of negative selection. Some do not and are important for T cell migration and other functions. But this picture changes again in the involuted thymus with the emergence of these AA-Tex1 and AA-Tex2s. Now we still have that normal MTEC differentiation, but there's many arrows now extending from these AIR-negative medullary thymic epithelial cells into the AA-Tex1 and AA-Tex2 populations, suggesting that these cells give rise to AA-Tex, and maybe that's consistent with where they're localized. We tend to see them around the thymic medulla. Okay, so next we wanted to understand whether AA-Tex might be associated with this impaired regenerative capacity of the involuted thymus that I introduced, that second question that motivated this study. Now, the thymus retains substantial capacity to regenerate following acute stressors. These are things like severe infections or pregnancy or chemotherapy, other stresses. However, this capacity diminishes with age, and one way we can model thymic regeneration is to challenge mice with a sublethal irradiation. It causes acute thymic atrophy followed by regeneration. If we just assay the number of cells you can get from a thymus in one of these time courses here, in the blue lines we're showing that we get from a young mouse, and you can see that after this initial atrophy, within about a week the thymus begins to regenerate. And that happens very quickly, such by about 4 weeks of age, it's approaching near normal levels. In the red lines, though, is the data from an 18-month-old mouse, and you can appreciate that it lags well behind its young counterparts, and some of these never quite get back up to normal steady-state thymopoiesis again. So Kaylin decided to do light sheet analysis again to identify what's happening with these AA-Tex microenvironments, and she chose day 28 as a good time point because you can see here there's a big difference in the regenerative capacity of the old thymus compared to the young ones there. So the image on the left is a thymic lobe taken from the involuted thymus of an 18-month-old mouse, and we're highlighting these AA-Tex regions in cyan. And then on the right is a lobe taken from a mouse 28 days after irradiation. You can appreciate that it's still somewhat smaller, consistent with that impaired regeneration, but it also appears to have a greater volume that's occupied now by these AA techs, shown again in cyan. This impression is borne out by light sheet analysis of various time points, and we can see the overall thymic size remains about half normal at day 28, and this is driven by the loss, particularly of the medullary region, but also the cortical areas. You can see in this quantification that the area taken up by A-tech is about doubled, and this represents an increase in the total number of age-associated techs after this damage model. We conclude then the expansion of AA techs correlates with impaired thymic regeneration. We also found evidence that AA tech take an active hand in this impaired thymic regeneration. So this is some data representing cell chat analysis, which predicts major signalling inputs and outputs being shared among cells based on the single cell transcriptomic data. And this is on the basis of things like receptor-ligand pairs, but also transcriptional evidence of cells receiving signals through these receptors. And the way to look at these plots is that we've arrayed all the various thymic stromal populations on the outside. So all the thymic epithelial populations are kind of on the top half of this, and including fibroblasts and endothelial cells that are arrayed on the bottom. Now, Anastasia took cells from mice that have been sublethally irradiated, and 7 days afterwards, so just as they're beginning that regeneration phase, performed this cell chat analysis. And she focused on signals that are known to be important for driving thymic epithelial cell regeneration. So FGF signals, KGF and others are really important. And you can see in the 2-month-old thymus, the fibroblast and mesenchymal populations are sending FGF signals into these thymic epithelial cells to power that regeneration that occurs in the young. But this is skewed when in the aged thymus, where you have the presence of AA-Tec1s, shown here in this cyan colour, and AA-Tec2s in that mustard colour. And so you can see that many of those arrows that were going to those normal thymic epithelial cell populations, the medullary ones, have been skewed now in favour of the AA-Tecs. It's the same story with another trophic signal. This is BMP signalling that typically comes from endothelial cells to drive the differentiation of thymic epithelial cells. Again, many of these arrows now are skewed towards the AA-Tec that are receiving those signals at the expense of the medullary thymic epithelial cells. So consistent with the situation where the AA-Tecs are now not just simply not supporting T-cell differentiation, but they're co-opting those reparative cues that are important for driving regeneration after damage, and perhaps explaining why the involuted thymus is no good at regenerating. So to summarise this bit, age-associated techs lose the capacity to support T-cell differentiation and show features of partial EMT, and they may be derived from air-negative medullary thymic epithelial cells. And importantly, age-associated techs expand following damage and appear to co-opt these regenerative factors. And so some of the implications of this work are, it first of all identifies an overlooked aspect of thymic involution that might shed light on processes that underlie this loss of function. And if we come back to those propositions about why the thymus involutes, it's tempting to speculate that if involution is an active process to conserve energy or to prevent autoimmunity or something, then perhaps that's a major function of AA-Tecs. They take an active hand in this process, and in fact they are there to induce thymic regeneration, thymic involution. And then finally, an implication is if we can work out ways to ablate or reverse these states, it might open new ways to regenerate the thymus and potentially restore immunity. Still much to learn. And so then I'll switch to the second part now of the talk, where we'll leave the thymus and deal with another cell type that's essential for immune tolerance. As we touched on earlier, a key process in the thymus is this randomization of the TCR specificities. And it's followed by this Darwinian selection process to try and whittle down the repertoire to those that are useful and safe. However, the process has flaws, and autoreactive cells can emerge into the peripheral immune system, and this is represented here by this black clone that's sneaked into a lymph node here. In addition to this leakage, there are a multitude of other things that we need to tolerate, but really would stretch the definition of what self is, at least that can be represented in the thymus. These are things like commensal organisms that we need to digest our food, dietary factors, things we inhale, and even the developing fetus. We need tolerance of these factors, because if that should break down, what happens is these autoreactive T cells become activated. They make many copies of themselves and then move into tissues where they can exert their inflammatory functions and cause chronic inflammation. About 20 years ago, it became apparent that there's another important mediator of tolerance to these factors, and it's entirely separate type of T cell that arises in the thymus, and it can also home to lymph nodes and move into tissues. And their job is to police the function of these conventional cells and stop them from wreaking havoc. These are referred to as regulatory T cells, or Tregs, and they use a variety of mechanisms to either prevent the activation of autoreactive T cells in lymph nodes or prevent their activity in the organs if they make it to the tissues. Now, to carry out these functions, Treg assume a number of distinct features. One is that they can routinely settle in non-lymphoid tissues really early in life. They're there to kind of anticipate autoreactive T cell responses, if you like. And then another is that they have a really high rate of turnover. Up to 10 to 15% of these cells in the spleen and lymph nodes turn over and are replaced every day. So this very high rate of turnover prompted us to explore how Treg homeostasis is controlled, and with the notion that if we could identify differences between the homeostatic control of T cells and regulatory T cells, then maybe we could take advantage of these distinctions to treat diseases. The promise of Tregs is that they impose a dominant form of tolerance. So you don't have to kill off every autoreactive T cell. If you're able to impose tolerance to them through regulatory T cells, that's a very attractive way of trying to treat disease. In previous studies, we found the intrinsic pathway of apoptosis plays a critical role in this process. Removal of certain pro-apoptotic members of the BCL2 family of proteins led to abnormally high levels of regulatory T cells in lymphoid tissues, and the removal of MCL1, a pro-survival protein, but not the related proteins BCL2 or BCL-XL, that led to a decrease in regulatory T cells, and the mice got really sick because they couldn't maintain immune tolerance. This mechanism was primarily controlled by IL-2. IL-2 is a key growth factor for conventional T cell activation. Tregs are very good at stealing IL-2 away from conventional cells, which not only prevents their activation, but allows the regulatory T cells to survive and thrive. Now, there's another pathway to apoptosis, the extrinsic or death receptor pathway, and following ligation of certain TNF family receptors, a signaling cascade occurs that leads to the activation of caspase 8. This is an initiator caspase that can then cause activation of downstream caspases that mediate apoptosis and dismantle the cell. In previous work, Karras made genetically deleted caspase 8 from regulatory T cells and found that the numbers of a certain class of Tregs went up in lymphoid tissues, in the spleen and lymph nodes, indicating that under steady-state condition, there's a different aspect of Treg homeostasis that's controlled by death receptors. We were curious to know whether this division of labour between the intrinsic pathway and the extrinsic pathway might change in inflammatory situations, the kind of settings that you would want to intervene and use regulatory T cells to treat. Perhaps IL-2 becomes less important, but these death receptor signalling becomes much more important. So to address this question, Karras collaborated with Mark Pellegrini and Simon Preston to challenge mice with lymphocytic choriomeningitis virus, or LCMV. This is a murine pathogen. It's much loved by immunologists because it establishes a chronic and really overwhelming viral inflammatory response in the host. And in black, we're just showing that while control mice are able to tolerate this infection, it's established within a few days but is maintained in these mice over long term. To our surprise, these conditional knockouts, the ones with FOXP3 Cre to delete caspase 8 only in the regulatory T cells, succumbed to pathology and had to be euthanized. The reason for this was an overwhelming inflammation that was manifest in immune infiltration in a range of organs. In the control, in the top here, you can see there's minor inflammation, but in the conditional knockouts, there's this severe inflammation, particularly the exocrine pancreas, some inflammation of the salivary gland, kidney, some liver and liver damage, and pneumonitis in the lung. Now, the reason for that is that the regulatory T cells were now disappearing. So the top panels here show a quantification of Tregs in the spleen, typified by the expression of FOXP3, a key transcriptional factor for regulatory T cells, and the TCR. And you can appreciate, if you compare control with the conditional knockouts, there's a proportional increase in regulatory T cells and a numerical expansion of about threefold more Tregs than normal. That's because this apoptotic process is not occurring anymore. But in the LCMV infected situation, this reverses, and now there's a loss of regulatory T cells at both the proportional and numerical level. And what that does is allow for hyperactivation of conventional T cells now, and that's what's mediating this inflammation in a range of organs and causing the death of the mice. So you'll recall from James Murphy's excellent Institute seminar just a couple of weeks ago that caspase-8 is at the centre of another form of cell death called necroptosis. In this situation, caspase-8 has pro-survival functions. And this is because in scenarios where normal death receptor signalling is inhibited or blocked, for instance, by certain viral inhibitors, this backup process referred to as necroptosis is engaged. Without caspase-8 activity, you get persistence of upstream signalling, leads to phosphorylation of the pseudokinase MLKL, and that forms pores in the plasma membrane, leading to this leakage of cytoplasmic factors, and it resembles necrosis. And this is a major alarm signal for the immune system. And curiously, we noticed that regulatory T cells already have a very high expression of MLKL, distinct from what you see in conventional T cells, which have very little. So Tregs appear to be poised to undergo necroptosis. We're going the wrong way. So Karas set up an experiment now that added a compound mutant now, so these caspase conditional knockouts joined together with MLKL knockouts to ask the question whether if you blocked necroptosis, would that restore these defects? Now, Karas and Simon measured many things, but a key parameter is shown here, and that's the ratio of regulatory T cells to activated CD8 T cells or CD4 T cells, these two conventional T cell classes. In control mice, they maintain a relatively high ratio, one that can preserve immune tolerance despite the chronic LCMV infection. The loss of Tregs in the conditional knockouts, however, shown in the black bars here, perturbs this ratio, and now you have less Tregs and more activation of conventional cells that leads to that autoinflammatory pathology. When you knock out MLKL as well, though, you correct this. You bring it back to that sort of normal ratio of Tregs to activated cells, indicating that tolerance has been restored. And indeed, when they compared the survival of mice over time and did a histological survey of their organs, they found that the double knockout, the caspase-8 and MLKL double knockout mice were now healthy and could tolerate the infection. Now that's interesting because it indicates a switch. Recall that in the steady state situation, you had an excess of regulatory T cells because death receptor signaling is required for apoptosis. So we're interested to know whether this same switch occurs in regulatory T cells from humans and whether they have the same sensitivity to necroptosis. And for this, we employed a small molecule inhibitor that's known to be a good inhibitor of caspase-8, imricasan, and performed a dose titration in T cell populations that were purified from human blood. And here's the data for regulatory T cells, and you can observe this dose-dependent drop in the number of regulatory T cells that's been quantified here from a number of donors. And if we look at conventional naive T cells, we find that they were completely resistant. If the conventional cells had been activated, though, and had an effector or memory phenotype, it changed slightly. The CD4 conventional cells were still resistant to imricasan. However, there was some sensitivity in the CD8 cells, such that they were killed off at higher doses. We could confirm that imricasan was inducing necroptosis if we took a dose that killed regulatory T cells but not the conventional cells, and added an inhibitor of RIPK1, one of the critical components of the necroptotic pathway. We could rescue the regulatory T cells from death via imricasan, indicating that Tregs from humans are also highly sensitive to necroptosis. So to summarise this quickly, inflammation causes Tregs to become highly sensitive to necroptosis, which can be pivotal in immune outcomes. MLKL is regulated by distinct mechanisms in Tregs versus conventional cells during viral infection, and also T cells from humans are highly and selectively sensitive to necroptosis induced by imricasan. So this prompted another question then: in what context might Tregs be poised for necroptosis? Because of the presence of this switch, it promises the ability to fine-tune regulatory T cell number and function in different settings. And so in this last little bit, I'll just quickly skip through that Lucille Rankin now has done a survey of regulatory T cells from a range of different organs, because you might recall before I said Tregs reside in a range of different non-lymphoid tissues where they can prevent autoimmune responses. What's becoming clear is that they assume different transcriptional processes and adapt to their tissue microenvironments. And it can also have impact on non-immune functions. Things like metabolism, thermogenesis, and tissue repair are all key functions that regulatory T cells in these tissues can have. And they mediate this by conversations with macrophages and stromal cells in those tissues. Various transcription factors in Tregs can direct their fate and their ability to mediate these functions in different tissues. And GATA3 is a really prominent one in this regard. Now, it's a little more complicated in the gut because there's commensal organisms and dietary antigens that we need to tolerize there. And there, another form of regulatory T cells, these raw gamma T positive Tregs, arise in the gut. And they seem to be linked to other functions such as influencing the composition of microbiota, nutrient availability, and neuronal function in these tissues. So when Lucy did a survey of these conditional caspase-8 deficient mice, looking at Tregs, and she did a huge amount of work here. I'll just highlight the main finding, which is this GATA3 positive Treg population was selectively lacking in the adipose tissue, small intestine, and colon. They were present in normal numbers in the lung and other tissues, but for some reason, they were particularly relied on caspase-8 in these tissues. As she compared now in the gastrointestinal tract, where we had at least these two flavors of Tregs, the GATA3 positive ones and the raw gamma T positive ones, she found that indeed it's the loss of these GATA3 positive Tregs was really obvious, but the raw gamma T positive ones were preserved. And we knew that this was being mediated by a necroptosis because if you took the compound mutant that now lacks MLKL required for necroptosis, that restored the number and proportion of these GATA3 positive Tregs. To understand why this might be the case, we looked at MLKL expression. I'll just highlight the take-home message here is that specifically this population of GATA3 positive Tregs that express KLRG1 have high levels of MLKL compared to the conventional counterparts or these other Tregs that can be identified in the tissues. For a variety of reasons, we settled on the question of whether a key cytokine required for GATA3 positive Tregs was mediating this upregulation of MLKL, and this is IL-33. So what Lucy did here was she sorted regulatory T cells, bathed them in IL-2 to keep them alive, and incubated them also with IL-33. And in some cultures, we removed IL-33, kept it in others, and then sorted the various Treg populations to see whether IL-33 could induce MLKL. When it wasn't present, neither of these populations were able to upregulate MLKL. When IL-33 was present, these ST2 positive ones, which are the GATA3 guys, it upregulate MLKL, but not the ones that don't bear the receptor. But we know that these ST2 negative ones can upregulate MLKL because if we provide type 1 interferon, which is known to upregulate MLKL, that can still happen, and that's just quantified here. So we have this situation where even in the same tissue, one subset of regulatory T cells have this very high sensitivity to necroptosis, the GATA3 positive ones, but their counterparts, the raw gamma T guys, are not, in part because of this differential regulation of MLKL. So to understand the consequences of this curious phenotype, Lucy turned to the most common model of colitis in mice that's employed, and that's using dextran sodium sulfate in the drinking water of the mice. And if you do it under a repeated administration, you establish this chronic colitis in the mice. And we wanted to know whether the loss of these GATA3 positive Tregs in the conditional knockout mice might influence the outcome. It did, but in a way we didn't expect. So you can see in the black lines that control mice lose weight during the DSS administration, but then they quickly recover it until you put it in the drinking water again. The caspase-8 conditional knockout mice shown in red, though, you can appreciate lost less weight, and when they recovered, they came to a much higher baseline than the control mice, indicating that they're able to tolerate the DSS much better than the controls. And this was strange because what we felt that the Tregs would be important for is the inflammation in the tissue, but that's not what we found. If we measure inflammation in the colon, either by colon length, it normally shrinks if there's inflammation, or a histology score done in a blinded fashion by Tracy Petoksky, thanks for that, we found that there was no difference in the inflammation in the colon induced by DSS in this model. And so it seems that the regulatory T cells, the GATA3 positive Tregs, are important for some other factor, and we suspect maybe it's this metabolic regulation that's required in fat and other tissues that might be controlling this differential response. So to summarise this bit, the GATA3 positive Tregs residing in tissues are especially sensitive to necroptosis under steady state conditions. Again, remember the contrast in the normal lymphoid recirculation, regulatory T cells are increased in number when we inhibit caspase-8. This is driven by IL-33 mediated MLKL expression. We believe there must be other cues present that derives this differential sensitivity. And the GATA3 Treg necroptosis counterintuitively improves metabolic outcomes in this chronic colitis model. And some of the implications of this work is it might inform how to design cellular therapies with improved in vivo persistence. And it highlights a distinction between regulatory T cells and their conventional counterparts that might be targeted to relieve immunosuppression under certain circumstances where it might be advantageous, for instance, in cancer. And so with that then, I'll finish off. Here's some of the acknowledgements. I've underlined the key players as I've gone through, but I just want to highlight a fantastic collaboration with Kelly and Verena, the CDI that helped all that imaging work at the very beginning, Mark and Simon for the pathology models, and Jared and Marcel Vandenbrink, our collaborators in the States, on some of that thymus work. And finally, we're supported by great support services throughout WEHI, but I just wanted to highlight the bioservices team that have looked after our mice for so long. They're central to all of our studies, and they've had to work under really challenging circumstances over the last few years, so hats off to them. And with that, I'll take any questions there might be.
We have time for some quick questions.
That clock is 4 minutes fast, by the way. So.
Could I ask any thoughts about...
You'll need to turn on the mic. Give it a sec. Just shout then.
Yeah, great seminar. Question about the inflammation of Tregs and necroptosis. Has this been looked at in transplant settings such as the graft versus host disease?
So Ash is asking whether Treg necroptosis and the inflammation has been looked in graft versus host disease. I don't believe so, in part because the reagents to look at that, or at least in people, don't exist, except James and his team now have created some really excellent tools. So maybe IBD is a place for us to look at, because one proposition based on James' beautiful data that he presented in his seminar is if necroptosis is contributing to IBD, the fact that regulatory T cells in those tissues are highly sensitive to necroptosis might suggest you get a bit of a feed-forward loop there, where you're losing that immunosuppressive capacity of Tregs because they're undergoing necroptosis, and that leads to worsened inflammation. Angus.
Castration. Castration delays or improves involution. How does that impact your AA test?
Great question. We're investigating it right now. So castration, so removal of sex steroids is a great way to induce regeneration of the thymus in aged animals, including humans. It's transient, though, it's a chemical castration in that case. We have preliminary data, and Michael Dandria in the lab has done this, indicating that it's opposite. So when the peak of regeneration after sex steroid ablation, AA techs are gone, mostly gone. That interests us because one of the things that distinguishes them from all the other stromal cells, they have really high expression of androgen receptors and estrogen receptors. And the fact that sex steroids are inhibitory for thymic function suggests to us that maybe this is an important player. This is part of the reason that AA techs are there, and that that suggests why there's a different model. If we take sex steroids away, that's a way to potentially redeem AA techs. Great question.
So, great, I'm just wondering what are the goals of AA tech during homeostasis, and what if you read the AA tech from the start?
We would love to do that experiment. The problem is when you're dealing with thymic involution, as soon as you have an idea, it takes a year or two to make the mouse and do it. We're exploring ways to try and deplete AA techs in chemical ways, but we're not quite there yet. But so because the gene profiling doesn't tell us what they are doing, I think the most compelling hypothesis is that maybe what AA tech are doing is causing thymic involution. Perhaps some of those propositions were right, that involution is important for a healthy immune system, even with age. But if we can understand what that negative impact is of AATEX, we can reverse it in those settings like bone marrow transplant where you want to get T cells back.
The last question from online. Can you follow thymic involution in human peripheral blood, and would infection be expected to increase the rate of involution? It's an anonymous question.
You can track thymic output in the blood. You do it by measuring these T cell receptor excision circles. So these are little bits of circular DNA made when that recombination process happens only in the thymus. And so that graph I showed you at the beginning about thymic involution was measuring those tracks. So you can measure it in those people. And yes, it is true that infection, particularly HIV and other infections, reduce that even further. So consistent with the idea that not only does the thymus involute, but it's highly sensitive to those acute stresses by infection and others. So incorporating TREC analysis, which in fact has become a newborn screening tool for inborn errors of immunity in the States, can be a really powerful way of measuring thymic output.
Okay.
And Australia. That's great news. Excellent. Okay. Thanks, Charlotte.
Well, please join me in thanking Daniel for a wonderful overview of new insights.